So extracellular matrix, the basic components of extracellular matrix are structural fibers like collagen and elastin. Hydrated matrix are made up of proteoglycans. Adhesion molecules are laminin and fibronectin. They behave as adhesion molecules. So basically, they provide structural integrity and protection to the plants, and they are providing making the cell intact, and they are helping in movement of other components also inside the cell. So inside the cell also as well outside the cell, and transport of molecule from cell to cell. So extracellular matrix play important role in transportation as well as protection and providing structural integrity to the cell. So basement membrane, they are behaving as support structure for resident cells. They are located beneath the epithelial and endothelial cells. Structural fibers are of two kinds. One is collagen and elastin. Proteins give shape, and they are soluble proteins like fibrous and globular proteins. Some fibrous proteins are not soluble, but globular proteins are soluble structures. We can check the solubility of proteins on the basis of their shape and solubility at 25 degrees centigrade, that is room temperature. So in vertebrates, the most abundant type of protein present is type collagen type 1, type 2 and type 3, and also secretory proteins. They are inelastic in nature. Collagens are such kind of proteins which are inelastic in nature, and there are 27 different kinds of collagens. So more than 90% of collagens are of type 1, type 2 and type 3. Collagen molecule is a polymer of tropocollagen actually. So its fundamental unit is tropocollagen. So collagen molecule is long and elastic. It is a structural unit. It is triple stranded, means tropocollagen is making a triple stranded structure, and it is forming into collagen, and it is right-handed helical structure. So the three helices make the right-handed helix form, and they bind in a helical structure to make collagen fiber. Collagen are having three polypeptide chains, and that are alpha chains. So it's a homotrimeric structure which is having three identical alpha chains, or it can be heterotrimeric structure having two or three distinct alpha type chain. Alpha chain versus alpha helix. So there are two terms. One is alpha chain and one is alpha helix. So what is the difference between alpha chain and alpha helix is that there are 3.3 amino acid in per turn in alpha chain, while in alpha helix they are having 3.6 amino acid residues per turn. If we talk about alpha chain, more than it is made up of more than 1,000 amino acids. In vertebrates, there are 25 different kinds of alpha chain that is coated by different type of genes. It is formed by membrane-bound ribosomes. It is having three alpha chains, right-handed triple helix structure. It is having left-handed helical structure. Each one is having left-handed helical structure, while it is the formation of the structure formation by the three alpha chains is a right-handed helix. They are having 3.3 amino acid residue per turn, where there is glycine, X and Y. The Y structure can be proline or hydroxylysine. So X is often proline. So there are three types of amino acid which are constituents of the turn, and it can be glycine and then X and then Y. X and Y can be any amino acid. X can be proline and Y can be hydroxyproline or hydroxylysine. So it helps in post-translational modification. Post-translational modification actually occurs in endoplasmic reticulum, and it is the process in which hydroxylation can occur. The hydroxylation occurs of mainly amino acid residues, proline and lysine, which to which we can modify the to which we can attach a hydroxyl group, and the other one is glycosylation. Vitamin C can act as cofactor for post-translational modification. It can results in formation of interchain hydrogen bond, and deficiency of which can cause scurvy. That is why vitamin C deficiency causes scurvy. Um, the hydrolysation of proline and lysine is caused by prolyl hydroxylase and lysine hydroxylase. Glycosylation occurs in endoplasmic reticulum. There are two types of glycosylation. One is O-linked glycosylation and one is N-linked glycosylation.